Good morning, everyone, and welcome to the Human Longevity Incorporated Media teleconference call. All participants will be in a listen-only mode. Should you need assistance, please signal a conference specialist by pressing the star key followed by 0. After today's presentation, there will be an opportunity to ask questions. To ask a question, you may press star and then 1 using a touch-tone telephone. To withdraw your question, you may press star and then 2. Please also note today's event is being recorded. At this time, I'd like to turn the conference call over to Mr. J. Craig Venter. Sir, please go ahead.
Thank you very much. This is Craig Venter. We're pleased to be announcing this morning Human Longevity, Inc. We're launching to promote healthy aging using the latest, most advanced techniques in genomics and stem cell therapies. I'm here with my co-founders, Peter Diamandis, who's an MD. He's a pioneer in the field of innovation and incentive competitions. He's chairman and CEO of the X Prize Foundation. He's co-founder and executive chairman of Singularity University, and he's a best-selling author of Abundance, The Future is Better Than You Think, and he's also a graduate of MIT and Harvard. My other co-founder is Bob Harari, who's one of the visionary scientists that helped shape the field of stem cell therapeutics. He'll serve as co-founder and vice chairman at Human Longevity, Inc., and he'll be expanding on more than 25 years of his work and deep expertise in stem cell biology, and the goal is to help restore lost functions in defective and aging individuals. Also with us this morning are two of our scientific and clinical advisors, David Brenner, who's the Vice Chancellor for Health Sciences at UC San Diego and Dean of the Medical School, and Scott Lipman, who's head of the Moores Cancer Center. Also with us is Karen Nelson, who's the president of the Venter Institute, and is the first to publish a paper describing the microbiome using high-throughput sequencing. So we're all pleased to be with you this morning. So we're announcing Human Longevity, Inc., and with that, we're building the largest human genome sequencing center in the world using the latest in Illumina technology. We've acquired 2 of their new HiSeq 10x sequencing systems. And as many of you know, sequencing has gone a very long way in the last 13 years. 13 years ago, it cost approximately $100 million and took 9 months to sequence my genome. Today, the cost of sequencing due to this tremendous technology change is down to about $1,000 a genome, and we're scaling up to do tens of thousands of genomes in the same time frame that it took to do one. But I've argued for a long time genomics is only a small part of the picture, even though it's an important one. And what's unique about what we're doing is we're combining human genome sequencing with sequencing of the microbiome, and we're going to be adding on multiple other layers of data, including the metabolome, which is all the chemicals associated with the human body. We're announcing an agreement with Metabolomics in North Carolina to characterize 2,400 chemicals in the bloodstream of 10,000 of the initial patients, to look at layering on the chemical data with the microbiome data, with the human genome data, and most importantly with the human phenotype data. We'll be importing clinical records of every individual who we're sequencing, so this will be one of the largest data studies in the history of science and medicine, layering hopefully within 5 to 10 years of having 500,000 to 1 million human genomes, the associated clinical data, phenotype data, outcome data associated with that. And we will be using that hopefully to make numerous new discoveries on preventative medicine, how to predict medical outcomes. We think this will have a huge impact on changing the cost of medicine. And also the human longevity is a focus on a broad human health because in order to have a long, healthy life, Generally, it doesn't work to do that if you have severe heart disease or cancer. And right now, if you reach the age of 85, you have an 50% or better chance of having a severe dementia. We need lots of changes in medicine, and we consider the announcement of Human Longevity, Inc. The first shot across the bow of changing the U.S. Medical system to a data-driven, fact-driven, information-driven system. We also plan to use this information along with stem cell therapy and techniques we've developed at my other company, Synthetic Genomics, to rewrite the genetic code of stem cells to correct errors and use stem cell therapy as one of the key adjuvants to changing outcomes long term. We're also announcing this morning a starting collaboration to start to change medicine with our colleagues here from UCSD, where we're trying to get genomics to be a central core of medicine. We will be doing up to 40,000 patients a year. We're starting first with the Moores Cancer Center, and the goal is to sequence the genome of every individual that comes into the Moores Cancer Center and to do full genome sequencing on their tumors. Cancer is one of the most actionable areas right now with genomic-based therapies. There's only a few that are truly actionable. We plan to extend that broadly across cancer, but cancer is just the first of a multitude of diseases that we'll be focused on and sequencing this year. We'll also be sequencing the genome of large numbers of everyone from infants to supercentenarians to understand and be able to predict the future outcomes in medicine. We'll be sequencing healthy supercentenarians that have somehow gotten to over 100 years of age with little or no disease, trying to understand the protective values of different parts of the genome to, as I said before, get into a preventative medicine paradigm. We have a stellar group of investors. Our initial capitalization, A round is approximately $70 million. We think that will carry us through the first 18 months as we build these operations. We have facilities already here in San Diego. We're building out the lab facilities, CLIA laboratories for this very high throughput microbiome and human genome sequencing. And we're building long-term laboratories that we'll be moving into in about a year, also here in San Diego. That's a quick synopsis of where we are. I would like to open it to Mr. Operator for questions from the call-in people.
At this time, we'll begin the question and answer session. To ask a question, you may press star and then 1 using your touch-tone telephones. If you're using a speakerphone, we do ask that you please pick up your handset before pressing the keys to ensure the best sound quality. At any time your question has been addressed, if you would like to withdraw your question, you may press star and then 2. Once again, it is star and then 1 to ask a question. Once again, in order to ask a question, please press star and then 1. And our first question comes from Brad Fikes from UT San Diego. Please go ahead with your question.
Yes. How many people will be on your payroll at first?
Well, Brad, thanks for the question. We are hiring rapidly. Our website at humanlongevity.com is live any minute if it's not live now, and we list a number of job postings there. We expect within the first year we'll be hiring over 100 new people to the San Diego area.
Our next question comes from Gary Robbins from San Diego Union Tribune. Please go ahead with your question.
Hi, good morning, Dr. Venter. What can you tell us about the investors, the $70 million, where it comes from, and the size of the labs you're building in San Diego County to do this project?
So we have a terrific investor pool. mostly very high net worth individuals. The lead investor is also the lead investor in synthetic genomics, and that's KT Lim of Genting Corporation. The actual entity name is Dragsac Limited in Malaysia. But we have a number of individuals. Illumina, another local San Diego company that makes this incredible technology, is also an investor in human longevity. The laboratories, we already have 2 laboratories of about 40,000 square feet each, and we're going to be building out a facility in UTC where we'll have another approximately 60,000 square feet for initial building.
And could I ask you to expand on what the sequencing of the microbiome is going to do here, how that's really going to advance things over where you are now?
I'm going to pass that to Dr. Karen Nelson.
Thanks, Craig. So, as you know, we've made a number of strides. Hello? Can you hear me?
Yes, I can.
So
yes.
We've made significant strides since 2006 in terms of understanding the impact of the microbes on human health and disease, and we're hoping that through analysis of these huge cohorts that we have access to, we can make real statistical correlations and start to push the envelope in terms of developing novel therapeutics and new probiotics. So that's also going to be an angle of HLI.
Thank you.
Our next question comes from Ian Jervison from DFJ. Please go ahead with your question.
Thank you. Yes, that was Steve Jervison from DFJ, and I guess full disclosure, happy to be an investor. Craig and team, can you tell us a little bit about where you see the earliest applications and how this might compare in scope and ambition to BGI in China?
So BGI in China, by the way, Steve, thank you and welcome to another company. We're
Absolutely. Happy to.
We're delighted to have DFJ with us, and you in particular. So BGI has set up a very substantial operation in China, and they have a fairly substantial number of much earlier Illumina instruments. My understanding, they have not upgraded those, and so these new instruments out of Illumina have substantially higher throughput capability. And in discussions with Illumina, they confirmed that our 2 10x systems would substantially exceed the capacity of the BGI in China. I think BGI's contributed a lot in the last decade to human genomics, but this is a non-saturable market. We have now about close to 8 billion people on the planet. And if genomics does what I hope it will do, where it becomes equivalent to a urinalysis and a CBC on admission to a hospital, we want everybody in this country to have their genome sequenced as the starting point in medicine. We can't have enough players in the human genome sequencing field. We're just trying to elevate it to a new level.
Thank you.
You had another question, Steve. I can't remember what it was.
Yes, right. Early applications. Where do you, in terms of sequence of how people will feel this in their lives, how do you think it'll play out and the scope of that ambition?
So our long-term goal is obviously getting to preventative medicine, trying to make predictions from the genome. An example that I've used with many before is having my genome on the internet. It's for 13 years. It's no secret. I'm heterozygote for ApoE4, which gives me a substantial increased risk for Alzheimer's disease. Having recent tests of brain scans with radioisotopes that measure amyloid in the brain, my brain came out completely empty of amyloid. There were other things in there, fortunately. And so the question is, and there's a large number of people, in my situation being heterozygote for ApoE4, that for some reason do not get Alzheimer's disease. which means there's protective alleles in the genome. And people are focused on disease genes. They're not focused on protective alleles. So it's true for breast cancer, for colon cancer, for heart disease. For every disease, there's disease causing or increasing risk alleles, and there's ones that seem to protect us. A big part of our study will be to find the protective alleles, which give a whole new approach for preventative medicine, even new drug targets, et cetera. So that's a longer-term view of getting towards healthy aging. In the short term, the reason we chose to start with the Moores Cancer Center is cancer is one of the areas that has evolved over the last 5 years of actually having actionable data from the human genome where it has predictive value for therapeutics. We have one of the leaders in the field here. Scott, would you like to comment further?
Yes, I mean, this it will be an unprecedented collaboration in the field of cancer and genomics, and to do full genome sequencing on all patients, solid tumors and leukemias that come through the UC San Diego Moores Cancer Center, not only of the tumor but also of the germline, and not just using the germline as a control for what's happening somatically in the tumors, but actually to look for new cancer predisposing genes and so on, and mutations. And in the field of cancer, as Craig just mentioned, the pace of translating discovery of these so-called actionable or genetic drivers to the clinic has picked up rapidly. And so I think this and in lung cancer particularly, with the discovery of the ALK fusion translating to standard of care within 4 years, the recent discovery of the RET fusion in lung cancer getting to the clinic in about a year. So the pace has really picked up, and with this level and scale of sequencing, this will really accelerate discovery of new actionable mutations in other tumors, like prostate cancer and head and neck cancer and others where they haven't been found yet, to get to the clinic quickly to help our patients.
David, do you have anything you'd like to add on that in general medicine? Or?
So, just briefly, this is a revolution in biomedical research. This is the first time ever that we as physician scientists have had the opportunity to handle very large data sets and to be able to compare the genetics of a patient with the microbiome, as Dr. Nelson said, with the metabolites in the blood, and to try to then gain new insights into understanding disease pathogenesis, into diagnosis, and hopefully into new therapies and cures. So this really is a tipping point for medical research.
Next question.
Our next question comes from Brina Fried from Clinimmune Labs. Please go ahead with your question.
Hi, could you expand on the involvement of stem cell therapy in this project?
So I'm going to pass that to Dr. Harari, one of the world's experts.
Good morning. So, as you can imagine, never before has there been an approach which aligns so many cutting-edge tools to interrogate the way the genetics of stem cells change during the process of differentiation and aging. A better understanding of that will enable us to identify ways to deploy cells in order to correct the accumulated defects that occur in the average aging patient's own stem cell population. So we see this as an important opportunity to validate the use of cell therapy as a means of addressing the changes that occur in aging. And as we identify the types of defects that occur, we'll be better equipped to design therapeutic tools to serve those defects.
Next question, please.
Our next question comes from Dan Malpe from NI Systems. Please go ahead with your question.
Hello. Yes. It's incredible what you're proposing, and I really wish you success. It's quite important. I'm curious on how you're going to handle the consent of the data as well as ways that you might share the data with others?
So we're going to be handling a huge amount of data. Obviously, we're sequencing tens of thousands of genomes potentially a month. We're working on all kinds of novel approaches, including looking at your approach, and I think we're talking to your company about some novel approaches. So in terms of sharing data, our goal is to accumulate and create the world's largest clinical, actionable, interpretable database of genomic and related information together with the clinical information. Obviously, doing this in CLIA-certified labs, the data needs to be protected and can't just be open on the internet because there's federal requirements and protection required for the patient's information. But we will be creating a unique log-on system for all of the physicians associated with the different hospitals. For example, the Moores Cancer Center and the scientists at the medical school here will have seats on our database where they can log in to get specific information. And we're working out how we will do that with other clinical centers. So it will be broadly available, but we must protect the individual's information. Scott, you want to add to that?
Yeah, so at the Moores Cancer Center, we're going to offer this to all patients who come and offer them consent for these kinds of studies. So all patients will be consented and offered this genomics.
Outstanding.
And our next question comes from Brian Clark from New River Community College.
Thank you, and it is an honor to speak with you today, Dr. Venter. I teach anatomy and physiology and also health, and my question has largely already been addressed, but I am in particular interested in the practical application of this, and I also have a particular interest in immunizations, and I teach a lot of that in my classes, and I'm interested in hearing a practical application as it relates to immunizations. For example, how you might work with a company like Inovio or some of the science out there in the immunization field.
Well, thank you for your question. I have tremendous respect for our community college system, having started my intellectual career at the community college before transferring to UC San Diego. So, I think I would refer you to my other enterprise, Synthetic Genomics, Inc., that has a very specific vaccine program based on synthetic genomics. I think what we're measuring and will be working on at Human Longevity will be supportive of others developing new vaccines, but won't be a direct goal of our company. Bob, you want to add to that?
Yes, again, thank you for that question. The other thing I might add is that as we are looking to better understand how cellular medicine might be applied to improve physioanatomic function in aging individuals, one of the obvious platforms we can rely on is using cellular medicine to improve immune function. And as you probably know, much of the work ongoing in developing therapies from stem cells has been in using their immunomodulatory and immunoactivation behavior. So that represents another possibility.
Next question, please.
Once again, if you would like to ask a question, please press star and then 1 using your touch-tone telephones. To withdraw your question, you may press star and then 2. And we have an additional question from Elizabeth Panisi from Science Journal. Please go ahead with your question.
Yeah, can you clarify how this will work in terms of the patients and the cancer center will give consent to have their genome sequenced and their microbiomes determined, and then what will happen to that data?
Liz, thanks for your question. I'm going to pass that to Scott. But let me preface it. We're still working out a lot of these issues. It's unprecedented to have it so patients more have to opt out of such an action, and we're doing that because we think it's so critical to change the face of medicine. But Scott and David are working on this diligently from the medical school side. So, Scott?
Yeah, so again, we'll offer this to all the patients after signing informed consent, and this will begin as a purely research, a comprehensive research collaboration to accelerate discoveries that will hopefully quickly get in to help our patients. And so, while this work is at the moment completely within the research realm, this initiative is anticipated to lead to transformative discoveries that will get to the clinic quickly. As I've mentioned, in lung cancer, that can happen within a year or so. So we're in this research collaboration. We want to better understand the development of cancer and its relationship to aging. And as you know, over the past few years, about 5 years, this type of work has really transformed the landscape of precision cancer therapy and personalized medicine. So that again, we're beginning with research, but we'll quickly move into clinical application.
But in terms of, if I understand from the press release, Human Longevity is going to build up this database of information that it's then going to provide, and I assume sell to pharma and other companies interested in this information. Is my understanding correct about that? And if it is, then isn't this going to be used for commercial purposes as well as research purposes?
David?
That's you.
Yeah, I think that clearly the objective here is to be developing technologies which will have a robust commercial value and commercial opportunity. And one of the greatest and unique differentiating factors about the HLI approach will be that it is focused on combining both a service business model along with the development of discrete products.
David Brenner?
I just want to add one thing. So the way you articulate is exactly right, so that researchers and physicians at UC San Diego will have access to all their patients' data, which can be used for research to advance biomedical research. At the same time, though, HLI will keep the data and can then combine it with many other data sources to get the largest collection of genomic and other types of data. So I think it's very synergistic. This is exactly what I would like UC San Diego to be doing, with its neighbors throughout San Diego.
And in that research realm, we're looking for new mutations, new translocations, new pathways that are altered with very strong bioinformatics approaches to discover new actionable events so that we can then move it into the clinical realm to help patients.
And I'd like to make it clear, cancer is just one of the areas, Liz, that
hold on. You need to identify who's speaking. I've heard three different people.
The last one was Scott Flipman. The one before him was David Brenner, and the one before him was Bob Harari, and this is Craig Venter speaking.
Okay.
So I just want to emphasize that, if we're going to do 40,000 genomes a year, we anticipate maybe 5, at most 10,000 of those coming from the Moores Cancer Center. We're discussing with David Brenner and our colleagues at UCSD in numerous other areas of medicine, unique patient populations that we think would benefit from this type of analysis and would benefit medicine in the long run. So while we're starting with the Moores Cancer Center because of the unique nature of it, in the end, even if we solved all of cancer, statistically, to quote Larry Page, that only changes human longevity on an average of 3.5 years or so. So we have to deal with medicine across the board and this information is critical, but it also emphasizes the individualized nature of this. And averages are totally meaningless to any individual. If you're the one with cancer, if you're the one with Alzheimer's disease or heart disease. So this is the ultimate individualized medicine program, but it only works if we have huge databases to get accurate, meaningful information from.
But I go back to my original question, which is essentially you're taking all these data from these patients and building a database which you're going to sell. Is that right? So isn't that a commercial use?
HLI is a commercial company because obviously this is a very expensive undertaking, and the only way to do that is to make it so the data is so meaningful that it does have commercial value. If it doesn't, this will be a very short-lived phenomenon.
And so will the patients be, when they do the consent, will they be told that their data will be sold and used?
They will be told that the data is part of the HLI database and they're getting their genome and their information in exchange for that. So we think that's a fair exchange. But those terms are still being worked out. Next question, please.
Our next question comes from Bruce Bigelow from Xconomy. Please go ahead with your question.
Good morning. Dr. Venner, I was wondering if you're going to have a role at Human Longevity specifically, and who's going to be running Human Longevity, and also I noted Peter's there, but I haven't heard from him, and I'm wondering what he's doing in this.
So, Bruce, nice to have you with us. Yes, I'm the chairman and CEO. I'm also the chairman and CEO of Synthetic Genomics, and I will be playing an equal role in both organizations. We have a phenomenal management team we're building at HLI, and we'll be introducing those to you and others in the not-too-distant future. I have an equally powerful one at Synthetic Genomics, and my two co-founders are co-chairmen and are providing tremendous intellectual guidance. But I will let Peter answer his own question.
And I'll just correct, Bob and I serve as co-vice chairman of the board.
Co-vice chairman.
Yeah. Bruce, I'm playing an active role with Craig and with Bob in the strategic growth, initially the capitalization of the company. We brought in $70 million in our seed round, which is a significant statement to the fact that there is a huge amount of interest here. This represents, one of the largest marketplaces in the world. There is, nearly 50% of our $7 trillion global health bill is spent after the age of 65 years old. There is some $50+ trillion locked up in living individuals over the age of 60 who would love to live longer. So this is one of the largest business markets on the planet. My role here is actively driving partnerships, innovations, working with the team here to grow the revenue streams and to turn this into a huge and profitable endeavor.
And Bruce, I'm sure you'll be visiting our labs in the not-too-distant future, so we look forward to that. So, operator, next question, please.
Our next question comes from Alex Philippidis from Genetic Engineering Biotech. Please go ahead with your question.
Hello. Thanks, Dr. Venter and everyone. I'm wondering if you can elaborate on the announcement speaks of initial clinical sequencing efforts on cancer in terms of the types of cancers and over what sort of time frame are you looking to expand into other areas, whether that's Alzheimer's or whatever other disease area?
Well, let me answer the second part of that first. And I thought I mentioned that earlier. Cancer will be a significant portion of what we do, but it will be a minority of all the sequencing that's done. And yes, we will be dealing with individuals with dementia, with autism, and hopefully a huge cohort of people that have lived a long time without disease. So we will be looking broadly across the disease spectrum. well-characterized individuals because the phenotype information is critical to this analysis. So a genome without the accompanying phenotype is useless information for all practical purposes. But Scott, you want to elaborate on cancer?
Right. So this is Scott Littman. We'll be sequencing or offering full genomic sequencing to every patient that comes to the Moores Cancer Center. So solid tumors, lung cancer, prostate cancer, and so on, as well as leukemias. And currently, obviously, it's a very active area in cancer, but most of the focus is on areas on targeted sequencing of known cancer genes, for instance, in lung cancer and melanoma. We'll be doing sequencing on all patients, and not just the 100 or 200 targeted genes, but the full genome, so 20-plus thousand genes on all patients, so that we'll hopefully be able to discover actionable, treatable genetic drivers in diseases that we don't have those in, for instance, prostate cancer, and discover new ones in lung. So it's very comprehensive.
Mm-hmm. Do I understand from your answer then you're looking more at sort of established areas, melanoma, lung cancer, any particular cancers?
No, we'll be doing every cancer. We'll offer it to every patient. I was just commenting that in the field, the use of target sequencing now tends to focus more on areas where there have already been shown to be some actionable mutations, like lung and melanoma. We'll be doing those, looking for new mutations, translocations, targetable or actionable events, but also other diseases like prostate cancer, head and neck cancer, where there are no actionable mutations, to discover ones to help treat those patients. So it's very broad-based in this collaboration.
Next question, please.
Our next question is a follow-up question from Brad Fikes from UT San Diego.
UT San Diego, yes. I'd like to find out more about the stem cell part of this initiative, especially whether the new Stanford Center for Clinical Genomics will be involved, and just what will the details of that be?
Bob?
Well, I think that undoubtedly this approach to looking at stem cells as a potential tool to address the changes that occur during aging will probably excite and enlist the collaboration and cooperation of most of the leading institutions in this field, as well as industrial leaders. So we're going to be very open and engaged with those partners in the space. Again, it is unusual to have this level of expertise looking specifically at the genomic changes that occur in stem cells, both to better understand the process of differentiation and specialization of stem cells, as well as how defects accumulate during aging and how those defects are associated with predisposition or onset of disease. So the institutions that you're describing are obviously perfect collaboration opportunities, and HLI will be very open to those.
Let me also mention that so Larry Goldstein is a major collaborator of my team at the Venture Institute on stem cell genome sequencing, and we've been pioneering that, and he's the new director of the Stanford Center, and Larry is also on our Scientific Clinical Advisory Board of HLI, and so we hope to have a very close interaction with the new center here, supported by Denny Sanford. So it's an exciting opportunity for us.
And it's Peter speaking. Let me just say to everybody on the call here, stay tuned for more announcements on the stem cell side.
I will.
Wow. Next question, operator.
Me too.
Once again, if you would like to ask a question, please press star and then 1. And we have an additional follow-up question from Elizabeth Panisi from Science Journal. Please go ahead with your question.
Yeah, will the centenarians that you're sequencing be the pool of centenarians that were collected for the X Prize?
Liz, that's a great question. It's certainly under discussion. The good news is supercentenarians aren't as rare as they used to be, and there's a lot of them in the world, and particularly in the U.S. So we're discussing with various groups that have collected groups of those, but we think we'll be looking at some novel sources as well. So all that's under discussion.
Thank you.
And gentlemen, at this time, and showing no additional questions, I would like to turn the conference call back over for any closing remarks.
Great. Well, thank you very much, and thank everybody for calling in and listening. We're very excited about this new opportunity. We view this as just the beginning, the starting point of this new field that some of us have been waiting for a very long time, following or sequencing the first genome 13 years ago. I would refer anybody to humanlongevity.com. We think there's a lot of information there you will find useful, to our press releases, and we look forward to this exciting set of developments, and we urge you to stay tuned. Thank you all very much. Thank you, operator.
And ladies and gentlemen, today's conference has now concluded. We do thank you for attending today's presentation. You may now disconnect your telephone lines.